I'd like to thank all of you in the audience for hanging on today. I know it's been a long couple of days, exciting couple of days. It's also great for me. I did my PhD and postdoc in Berkeley, so coming back to the Bay Area is kind of like a homecoming or a high school reunion for me. I get to see a lot of familiar faces, a lot of people who've mentored me along the way. So today we're going to talk about shale. And so what you should probably be thinking about right now is how does a native Californian find herself living in the middle of Ohio? And I can point to this drill rig as motivation. So this is my field site in West Virginia. I've studied the deep subsurface or the subsurface most of my career. And so this was a great motivation, this opportunity to sample the deep subsurface and understand microbial life. So the other question you might be asking yourself is what shale got to do with it? And we'll talk a little bit more about that. And hopefully by the end of this talk, you'll have a sense of where we're going. Okay, so the deep terrestrial subsurface. Most of what we know about deep life comes from marine systems. And that's largely because of excess. So in the terrestrial systems, we haven't had as much opportunity to sample. And so, as it was alluded to, I did my postdoctoral work here in shallow aquifer systems. And so in the subsurface world, we call these people soft rockers. What we're going to be talking about here is actually life in the bedrock. And so these are what we call hard rockers. And so the life habitat changes drastically as we move down with depth. And so in this soft rock habitat, or in this aquifer system, organisms live in wide open pore spaces. And so they have lots of opportunity for nutrients. There's water flow. There's genetic exchange. As we move deep into the bedrock, in shales especially, what really constrains life in these systems is the porosity. And so we're talking about really small pores. And if the pores were large enough to contain life, it's actually the pore necks or the pore openings that may actually, at a 9-Darcy scale, may prevent any sort of water or fluid or nutrient exchange. And so that's captured here. Here we are in the soft rock, lots of porosity, and down here in the deep zone. In addition, as we move deep, we're going to increase pressures and temperatures. So the habitat that I'm going to be talking to you about today is about 60 degrees Celsius, which is like the hottest day in Death Valley. Salinity is about 4 times that of the oceans, and pressure is about 3,000 to 5,000 psi. So all of the organisms native or introduced in these systems must be adapting to this environment. So the terrestrial subsurface, it's been alluded to contain a large amount of biomass, but we really, really know nothing about deep life in these zones. Beyond that, there's a lot of interest, obviously, in deep life today because of the economic viability they bring. And so this is actually the star is Columbus, Ohio, for those of you who don't know where Columbus is. And I really like this graph here on the right-hand side. I'll try to be an equal opportunist as we go through this, because it really speaks to how young this industry is. So I started at Ohio State in November of 2013, and you can see the contribution, one, how young the industry is on the x-axis, right? So this really this industry in the Marcellus Shale had kicked off in 2010, but you can also see in that short amount of time the contribution that this resource has had to the eastern seaboard. So in the United States and North America, hydraulic fracturing is occurring. And I think when I talk about this internationally, what we represent right now is guinea pigs for the rest of the world, both in terms of the infrastructure and the energy extraction and the management of these resources, but also in terms of the environmental risks. And so it's a really exciting field of study. Okay, so what is hydraulic fracturing? Essentially, hydraulic fracturing is exactly that problem that I alluded to, is this porosity issue. Because these shales are so dense, the gas is essentially trapped within them. And so we need to excavate and free this gas. And so essentially, you drill down into the gas, or into the shale matrix, was about 2,500 meters, and then you use water pressure. It's about 15 to 20 million liters that basically fracture open the shale. And so what it creates is these hairline fractures that essentially allow gas release into the system and recovery at the surface. I'd like to say in Ohio, we've actually got rid of open wastewater ponds in the last year, but that's not true for all states. So now we actually keep them actually in reactors. So change is happening, but it's happening slowly. What about microbial metabolism? Why do we care about it? To be honest, I think maybe industry, again, it's a young industry, is just now opening their ears to microbial metabolism. I care about it, and you should care about it, because there's a large risk of infrastructure damage. And actually, here's an example in the Haynesville Shale in Texas, where as a model, they put down pipe, steel pipe, and assessed the corrosibility in the system. So it actually turns out that we have a large risk of contaminants into freshwater aquifer systems because of this corrosion. Another reason why we should care about it is for energy recovery. We spend a lot of money trying to make these fractures and essentially blow open this rock. And if we can't control biomass, that's going to be essentially clogging those fractures. And then what I'm hoping to allude to today is this ability that we can actually use biogenic methane. If we understand the metabolisms in the system, maybe typically a well life has a well life of around 5 years. Maybe we can make this industry more sustainable and sort of an analog to what they've done in coal bed methane. And what I'll talk about at the very end is our recent data that I'm working with JGI on, and its access to new organisms in these pristine, trapped shales. Okay, so for all of you who've wanted on your bucket list to tour the I-70 in Ohio, today is your lucky day. So I have a lot of field sites that are all located within about 2 hours of us on campus. The first field site I'm going to be talking about today is actually one in Marcellus. So this is a hydraulically fractured system. So basically input fracture fluids go in, and then we monitor the fracture fluids over time for a year period. We use metagenomics to generate hypotheses about how organisms are living in this ecosystem. We then went to a totally different shale system, so they were deposited at different ages, they're at different depths, and that's in the Utica, that's number 2 on the list. And we test those hypotheses, and we actually try to cultivate the organisms to validate some of those hypotheses we saw in the first shale. And then the last project I'm going to talk about very briefly is the pristine shale. And so that's that drill rig I showed you at the beginning of this talk. And essentially there, what we're doing is asking, what does life look like before we fracture it? And so we're going to have the first pristine shale cores coming out. We actually have them in the lab. Okay, so for the first study, we're going to be tracking fracking. This is actually what happens. So we take fresh water, we pump it in. I don't do this, but the fresh water is pumped into these blender systems. In these blenders, sand, which is a proppant to hold open those fractures, is added, and chemicals are added, typically biocides, clay swellers, other things to kind of control the ecosystem. All of those are mixed and inserted downhole, and potentially biomass, right? Because this is something that we hadn't thought about, or in the industry they hadn't thought about, was the fact that they were actually introducing surface organisms into this deep subsurface ecosystem. So this is the wellhead at the top, and we're going to monitor an early flowback. So those are the fluids that are coming up to the surface right away, right after hydraulic fracturing. And then we're going to monitor the system into later times up to a year period. The first study was done by my colleague Paula Mauser before I got out to Ohio State, and what she did was a 16S approach. And so what she found was that when she tracked these 3 wells that were located near each other, that she saw the same kind of microbial communities. And she knew some of the end members, but she knew little about their metabolisms. So this is a perfect place for metagenomics. And so we basically took her samples from one well, and we filtered them in the lab. We extracted the DNA. You can see all the nice iron in the system. And then we ran it on a HiSeq in collaboration with JGI and with the DCO, Deep Carbon Observatory. The key here is that we wanted to reconstruct genomes. We wanted to be able to model input in terms of substrates and outputs in terms of metabolisms so that we could look at coordinated metabolisms in these systems. And so we spent a lot of time binning these genomes to kind of come up with these metabolic maps. I do want to state that you can see the kind of samples we're working with. They're high saline, high iron, high hydrocarbon. And so a lot of time went into method development. So if you find yourself working in those systems, we should definitely talk. We're happy to share all of our protocols. Okay, so the first thing I want you to take home is salinity. So shales are former oceans. And so these systems have a lot of residual salt. And so as this water gets injected, it's fresh water at the surface, gets injected into these deep subsurface reservoirs, what happens is it starts to equilibrate with the shale. And so you can see that the salt rises over time. And actually it becomes kind of brine-like at the last 200 days. And so if you're an organism that's going to adapt to this, you have to have some sort of mechanism for existing across a wide range of salt tolerances. And so again, here's our salt graph, just to kind of orient you. Here's our input. It looks like lake water for all intents and purposes. Early time points, we actually see a lot of variability in the genomes, which is consistent with what we saw in the 16S, but that's not the focus of today's talk. What I'm really interested in is what happens when the system stabilizes. So this 14-day period is highly variable, but after about 14, 15 days, the salt stabilizes. And what we see, we had a great introduction by Mary today about Halophiles. I have to get that comic, by the way, is that we get a very, very low richness ecosystem, at least in terms of the bacteria and archaeal members. And so all of these organisms are halotolerant, so they have names like Halanaerobium, Halomonas, Methanohalophilis. And so, so this is kind of what is happening in this ecosystem. The first question we asked was, are these native? I thought we would go into this system and sample fracturing fluids and come up with all sorts of novelty. We worked in the subsurface at Wrighton, and for those of you that followed that literature, we saw a lot of really interesting organisms. I was disappointed. These are well-known halophiles. And so then we had to ask the question is, are these coming from the surface and they just look like surface organisms? Are these unique? And so we could actually track back to the genome level, not only to the 16S level, and ask the question, is this genome found in the input? And the answer almost exclusively to all dominant organisms at the end is yes. So in this system, we're basically injecting organisms from the surface that are proliferating and blooming in the deep subsurface. And we know that they're growing. They're not just resistant to death because we've done actual cell counts. And now we're starting to do fish on these samples. So we've just recently wrote a review, and it's a great time to write a review when there's only about 9 papers in your field. So asking the question about how shared are these organisms across shales. And so those are those 6 key members I showed you here. Here are the different studies that have occurred across the United States. And there's all sorts of differences about when they're sampled, but the key I want you to take home is Halanaerobium, which was that orange big block on the last slide, which was dominant, is dominant and persists across every system we've gone to, except for one in Michigan, which isn't as salty as this. The say key members are found in many of the other systems, including the methanogens. So my lab looks at how carbon cycling links to methanogenesis. And so yes, we look at moose rumen, and we also look at shales. We look at wetlands too. And so we're really interested in how those ecosystems differ and the factors that structure methane production, how they're controlled. So when we wrote this proposal, I hypothesized that this would be a hydrogen-mediated system because that's the model in subsurface systems. And in fact, what I failed to account for was the salinity. And so actually, all of the methanogens that we've recovered in the system are all obligate methylotrophs. So they're not using hydrogen, and they're not using acetate. At least that's what we were inferring from the genomes. And so they're using methylamines, trimethylamine, dimethylamine, monomethylamine, which is just the amount of CH3 groups on, or they're using methanol. And so if we want to understand methane production, and biogenic methane production is something that we could maybe manipulate or enhance, then the question is the methylamines coming from the input material? Because then it's not a sustainable process. Or could it be somehow produced in the system de novo, and then that would be a really sustainable process? And so the first thing we did was mine the literature. Fortunately, in the last year or two, there's sites like Ficus where you can mine the chemicals going downhole. And so we found that many methylamines are actually quite commonly used as inputs in the input chemicals and additives in the system. Methanol, it turns out, is added at about 1%, is really highly abundant across fractured shales. The other option here is that it's synthesized biologically. And so when organisms grow in high salt environments, some of the adaptive strategies is that they produce osmoprotectants. And so the idea with the osmoprotectants is that it creates this, basically balances the osmotic pressure from the outside as well as the turgor pressure. One of the model osmoprotectants is glycine butane, which is a methylamine compound. So the first thing we asked is, did we have any evidence in our metagenomics for the capacity to synthesize glycine butane in the system? And what we found was that the methanogens, this is actually, this little guy in pink, is a bug that we call Frackibacter. It's a new genus that we have only found in shales. In fact, it was surprising to me because when we pulled out the 16S, we blasted it, and I've never seen this before, and I've worked in crazy parts of the tree of life. There was no sequence anywhere near it in the data set except for those found in shales. So that may be a target, maybe an indigenous organism, and only gets up to around 2%. And then we have other organisms that could potentially use compounds in the input fluids, like choline, and convert those. So the next question was, well, that's all well and good. Those are great cartoons. Do you see any evidence for this? And so that's where the EMSL JGI, I've learned the new acronym since I was here. It's called Ficus. That's where this proposal really came in handy for us. We wanted to sample the methylamines across time in these systems. And so I collaborated with David Hoyt, who does NMR at EMSL. He's actually here in the audience. And what we looked at was glycine betaine, this osmoprotectant. You can see it's not detected in the inputs, and as we get to the saline divide, it's detected in the compounds. So this was really exciting. Methanogens can use glycine betaine as a substrate, but our methanogens can't. But glycine betaine can be fermented to methanogenic substrates. The next thing we looked at was, what about the input fluids? Is that still a reasonable hypothesis that could be supporting methanogenesis? And the answer is yes. We see a large amount of methanol, as well as dimethylamine and monomethylamine going into these systems. And so it looks like a combination of factors. One, that we're basically priming the community, but two, that it could also be the potential to synthesize substrates de novo. So the catch here is that we're talking about an intracellular substrate. So glycine betaine is an osmoprotectant. It's inside the cells. So we had to ask ourselves, if we're detecting it at that kind of quantities, how much cell turnover is happening in the system, and what could be mechanisms for this cell turnover? And so we hypothesized that maybe there was a role for viruses. And so we thought that maybe prophage, that these viruses could be coming in with their hosts. We've now taken them from a surface environment, put them under pressure, put them under salt, doused them with heavy metals, and maybe we were inducing prophage, and then that could be triggering a lytic cycle that could be responsible for the release of betaine glycine. So when we started this project, and I still think this maintains today, we know very little about viral roles in the deep subsurface. Probably even the deep biosphere, or even the subsurface systems in general. Here's a study done by Engelhardt and Kallmeyer, where they just did cell counts in different marine sediments. And they saw that while viral particles decreased with depth, they were still much higher, 4 to 5 times higher than their biological or their bacterial hosts. Just at a genome level, we were surprised. When we started assembling these genomes, these were low diversity systems, high strain variability, but low richness in terms of the bacterial and archaeal communities. But what we were pulling out was about 100 or so viral genomes at each time point. And so we had about 300 viral contigs across the data set, and of those we had 21 closed viral genomes. So on par with what we were seeing in terms of closed genomes with the microbes. So we asked the question, can we actually start identifying a role for viruses in the deep subsurface? Is there a mechanism that we could actually link these viral genomes to their host genomes? And it turns out that the CRISPR system, so like you and I, bacteria have immune systems. And so the idea is that these bacteria that have this immune system, they essentially take a snapshot of the DNA, any foreign DNA, and they encode it in their genome. And so when these bacteria encounter that foreign DNA again, they have some resistance mechanism towards it. And so we thought, okay, well, we have a bacterial pool and a viral pool. Can we mine the bacterial pool for those viral signatures and then take those into the viral genomes and say, okay, well, we can at least say that this virus has interacted with this host. And so when this is done before in other systems, over 3-year studies that have really targeted viruses, which was not our focus here, they found 15 or so linkages. When we did it, we found a huge number of linkages. In fact, what we found more interesting too at some level was that every organism that was sustained or persisted in these ecosystems over time all had a CRISPR system. And then we could link those organisms. So what you're looking at here is the genome. So this is our host. And then this is the viral genome that matched. And you can see over time. And so we have basically an interesting case here with methanogens where we have one virus that is 2 different genera of methanogens. And so we can maybe, as we go through this system and sample more of these systems, we can study host viral specificity, which is something in viral ecology that we're really interested in. And this is just to show you that we are pulling out closed genomes. These just aren't contigs. And this is one that we could actually link to Halanaerobium, which is that dominant member in the ecosystem. All right, so we asked the question now. Now we're in one part of the I-70, and we're moving along the I-70 back into Ohio to a different shale system. But we were kind of armed with this information. We had inferred some metabolisms. And so now we actually went into a different shale with produced fluids and tried to culture these organisms. And so what you're looking at here, sorry, I promised I'd be an equal opportunist. What you're looking at here is 16S relative abundance data. And what you can see, this is actually what we recovered from our metagenomes. And what you're looking at with the red underline is the isolates we pulled out. And so we were actually pulling out isolates that were 99% similar to the 16S sequences from a totally different well. And all of these genomes, this is just a handful. This is halanaerobium ones because it was the most abundant. And here's our novel Fracibacter that's pretty low abundant, but conserved across systems. And all of these genomes are actually available in IMG today. So now that we have these isolates in hand, we were excited to ask more questions about them to support these metagenomic hypotheses. And my poor student who brought this data to me, whenever he grew the halanaerobium on rich media, it would crash out. And so you can see this here, right? It has this very short stationary phase. And I kept making him redo these experiments. And then finally I remembered back to this viral story. And so we actually went in and tested that hypothesis. Could we actually be inducing prophage in this system under this growth regime that we didn't see under other growth regimes in the lab? And so the answer was, yes, we could. And then we asked the question of which factors, chemical factors, could induce this lysis. And we'll talk about this in a second. But we tried all the standards, which is mitomycin and other things, and none of those induced the viral viruses in the system. But when we added succinate, which we saw was high in the HPLC, or we dropped the pH, or we added heavy metals, all things that could be happening in this ecosystem, we could actually induce the prophage and get the system to crash, the bacterium to crash. And so now we've basically sequenced the isolate, and now with the help of Matt Sullivan's lab at OSU, we actually now have the viral genome ready to submit to JGI. So we're really excited because now we can start doing some more experiments, understanding this relationship and what ramifications it may have for controlling population in shales. All right, so what shale got to do with it? Shale is the source of salinity in the system. It also is a rock-hosted environment. It houses the life. And so the salinity is what actually produces or controls the production of osmoprotectants like betaine glycine. We are hypothesizing that viral release is one mechanism that could release this betaine glycine. We've now then tracked other organisms and looked for organisms that can break it down. It turns out the only organism in the system in this fractured shale, along with 3 other temporal time series we have, that has the glycine betaine reductase that produces the methanogenic substrates is Fracibacter. And so we think that while it's low abundant, it may be a keystone organism for basically converting osmoprotectant to methanogenic substrates, which then are converted by the methanogens in the system. In this system, we are still getting potentially methane recovery from input fluids as well. And so as we extend our time series, what we're hoping to see is that methanogenesis and isotopic signatures of biogenic methane increase, while the methanol and those other substrates decline. And so this alludes to the fact that maybe methane enhancement could be a future target. So again, we just took this back to the lab. This is another Utica sample. It turns out we have Fracibacter in isolation, but we're having a really difficult time getting it to work or grow well alone. And so we said, okay, let's just go back to the drawing board. Let's just take fracture fluids or produced fluids and add these substrates and see if we can drive methanogenesis this way. And this is what you'll see. So here's TMA with no antibiotics, right? That makes sense. We're feeding the methanogen its preferred substrate. There's probably not a lot of other organisms it's competing with. But what we were really excited about was this glycine betaine. And so we actually have submitted these for sequencing, so we're excited to see who is there in that glycine betaine, and if it is our Fracibacter, that we couldn't get to do it alone in the lab, but maybe it needs support from other members of the community. It turns out this glycine betaine reductase is pretty sensitive to hydrogen and other things that maybe we just couldn't recreate in a pure culture. Okay, so I wanted to end with life in pristine shales. And so the Department of Energy, the NETL, has basically started 3 field studies. And so this is actually a great opportunity because for the first time, academia and industry and the Department of Energy are all sitting down at the table. And we're talking openly about chemicals. We're talking openly about methods for harvesting energy from the systems. This is the first one to kick off. It's in West Virginia. It's a Marcellus field site. We have another one kicking off in Ohio. It's in the Utica Shale. And then we have another one kicking off in Texas. And my lab is doing all the microbiology of these systems. So we spent the last year and a half actually developing methods for how do we, when we get these cores, how do we decontaminate them? And how do we ensure when we're talking about life in these systems that we're really talking about life that's pristine in these ecosystems? And so this is just a quick overview. This is basically, we're drilling 2,500 meters into the deep subsurface, and we're sampling at 7 different depth intervals into different formations. And this is one that's just sandstone that rests above the Marcellus at the top and the bottom, and you can see these cores coming out. Really quickly, we've submitted these samples. We just did quick PLFAs to see if there was biomass. So we've cleaned them, we've processed them, and what we find is the drilling muds, which is the input material contaminant source, have a very different signature than our cleaned processed cores. So at least that's promising that at least our decontamination procedures are maybe effective. What we also see is that there may be differences between the rocks. So this is a Marcellus and this is a Mahantengo, which is a sandstone shale located above. And so we're working with JGI. These are really low biomass samples. Not only that, biomass isn't low, but it turns out when you lyse cells in these systems, even when we spike it in, it all binds to the shale. And so we have some enrichments that have cellular life, which we're hoping will get high enough density to maybe do some single-cell genomics. And we're working to do metagenomics now. So that's where we are with this project. It's a really young project. Again, we just collected these samples in December, but we're really excited about this collaboration. So this is definitely a group project. A lot of the team originates at Ohio State. So it's my group and microbiology. A lot of the viral omics work and viral work was led by Rebecca Daly, who actually was a Berkeley transplant as well. It turns out the shale project was pretty enticing. Rich is the bioinformatician in my group. And then Mike Wilkins is in earth science. Dave Cole is another colleague in earth science. Shika's in earth science at West Virginia. And then Paula is in engineering. And together we kind of work and target, divide up the labor on this project. And so this work wouldn't be possible without support from the JGI. There's a lot of people listed up there. We're really excited. A lot of the low biomass methods have come from feedback from them, so we're not going into this blind. And then I've shown you the power of metabolites for really validating metagenomic hypotheses. And thanks to Tina Turner for inspiring the title talk.
Any questions? So this seems like a closed system, and if there is life there, how long ago did it get closed?
Yeah, so this is a great question, and actually one of debate amongst geologists is how closed is closed. So paleopasteurization, if we're assuming that life survived through that would be about 450 million years ago. But there's evidence that there's fissures along these bedding planes and that they might not be as closed as we originally thought. In fact, my colleague in geology, he's taking these fantastic images. And so it may be more heterogeneity in the system than we thought. And so it could actually house life. So.
Any other questions quickly? Okay, thank you once again.
Thanks.